Assalamualaikum warahmatullahi wabarakatuh. Hi, my name is Renata Ziza from semester 3, class A.
Petri dish or teleport petri is a container that is round, is shaped and made of plastic or glass. Used for cell culture, petri dishes are always in pairs. The smaller one is the container and the larger or is the light it is as container from trophy investigation and also from culturing bacteria, yeast, spora, or seeds. Plastic petri this can be discarded after single of for use bacterial culture. A porcelain dish. Every porcelain dish is a laboratory tool made of the porcelain which is a laboratory equipment material that is the resistant to heat. If we look at this tool, it has the same material as the corbel, but the shape and use of the porcelain cup is slightly different from the corbel. Porcelain discs are usually used to export the sulfide parents in the sample. A porcelain cup has a hemisphere is scale shape similar to a small bowl. This shape makes the evaporation process easier because of its wide surface area. You see, to rescue seminal substance in small amounts, the function of the porcelain cup is to reach substance at high temperature as the filter prepare and decompose the participant in the gravity so that it become a stable form. The thermopipette is one of the instruments assumed in the hemocytometer set package. There are two types of thermopipette with a difference in the glass grain in the pipette bulb. The thermopipette with red granula is the thermopipette from erythrocyte, while the white is the thermopipette from leukocyte. In addition to the glass grains, the dilution number are also different. At the end from the red color ending with the number 101, while the white color 11. However, test numbers are not absolute volumes. But the most important volume ratio is the dilution of the bulk that occurs in the pipette. Hematocrit centrifuge is a variation of microcentrifuge that serves to accommodate chemically sample from measuring the volume of packed cell hematocrit. Centrifuge is a device for rotating the sample at high speed, forcing the heavier particle to collect to the bottom of the centrifuge tap. Separation between filtrate and substrate the working participle of the centrifuge is the until the centrifugal force. All the centrifugal force is also exerted by the gravitational force. The faster the rotation, the higher the force generated. The main component is tau in the motor. In the motor there is a rotor and stator. If electric current is applied between the rotor and the stator, a magnetic field will arise electric. So in principle, the work of centrifuge is to convert electrical energy into mechanical energy in the form of rotation and base for the difference in the density of the mixed element in the liquid. Function basical centrifuge serves to the spread solid particular in liquid, for example to spread serum hematocrit, examination microscopic examination of urine. Centrifuge has several types, one of which is blood bank. Centrifuge is a tool that serves to spread serology sample and is commonly used in the blood banking and the serology. Especially, centrifuges are used for more specific purposes such as mixed raw hematocrit centrifuge and blood bag centrifuge, which are designed for specific use in the clinical laboratory. Micromatoric centrifuges are variations of micronutrifug and that can accommodate comprehensive sample for measuring the volume of hematocrit per cell with bulk. Bank centrifuges are the centrifuges used by blood banks and the serology designed to spread to serology in sample in tubs. Specialty centrifuges are used for more specific purposes such as mixed hematocrit centrifuges and blood bank centrifuges, which are designed for specific uses in the clinical laboratories. Micromatrotic centrifuges are variations of microcentrifuges that can accommodate complete sample from measure the volume of hematocrit per cell, while blood bank centrifuges are centrifuges used by blood banks and serology designed to spread serological sample in the tubs. An incubator in microbiology is an insulated and enclosed device that provides optimal conditions of temperature, humidity, and other environmental conditions necessary for the growth of organisms. The incubator maintains temperature, humidity, and other optimal conditions such as the CO2 and oxygen contained the atmosphere in it. The incubator is an essential laboratory equipment required for cultivating microorganisms. Under artificial conditions, incubator can be used for the cultivation of unicellular and multicellular organisms. The function of incubator is very important for many experimental work in cell biology, microbiology, and molecular biology. Laboratory incubators also have several types and of course also have different functions from one another based of on keratin parameter or the purpose of the incubator. The incubator is divided into the following types. Bentops incubator. This is the most common type of incubator used in the most laboratories. This incubator is the basic type of incubator with temperature control and insulation. Refrigerated incubator. For incubator at sub-ambient temperature use, the incubator is equipped with a modified cooling system with heating and cooling control. This type of incubator is called a cooling incubator. In the refrigeration incubator, heating and cooling control must be pressure balance. Check incubator. The dam technically controlled shaking incubator is another equipment used to cultivate microorganisms. Its advantages are that is the provided fast and the uniform heat transfer to culture faster and its aggregation profits in the cross aeration ratio in the access granule growth. However, this incubator can only be used from birth. Liquid culture media. Portable incubator. Portable incubator are smaller in size and are used in the fieldwork, egg environmental mix, raw biology, and water testing. Thanks for watching.